right, so in this last section, we're going to talk about the electron transport chain. This is the last step in cell respiration. So we've talked about glycolysis. We split the glucose into pyruvate. Then oxidation of pyruvate and Krebs cycle finished the breakdown of glucose into carbon dioxide. And it made a bunch of these coenzymes called NADH and FADH2. And those are the only thing that's going to go to our last step, which is the electron transport chain. So this occurs along the inner membrane of the mitochondria. So just a reminder, that is called the cristae. So let me just draw a little quick sketch of the mitochondria here. So just a reminder, this is our mitochondria. The Krebs cycle happened in the matrix. That was here in the middle. That's our matrix. That does not look like the word matrix. Sorry about that. And then the electron transport chain is going to happen along this inner membrane, specifically in the folds of the inner membrane. Usually they show it very folded like this. So within those folds called the cristae, that is where the electron transport chain is going to happen. And these folds are important because they increase the surface area, so there's a lot more membrane there by having it really folded than if it was just, say, this. If you had the outer membrane and this was the inner membrane, you wouldn't have nearly as much space for the electron transport chain as when if I redraw this with all the folds in it. Now I have more membrane, which means more reactions. Okay, so that's where this last step is going to happen, in the mitochondrial cristae. And what it's going to do is it's going to take the hydrogens and the electrons, because a hydrogen technically has one electron. And the electrons from the hydrogens are going to go down what's called an electron transport chain, which I'll show you in a diagram. And that's going to turn the hydrogens into hydrogen ions. So they're going to have a positive charge because they're missing their electron. So it turns out when electrons go down the electron transport chain, it generates power, energy. And that power is going to be used to pump these hydrogen ions, the H pluses, into what's called the intermembrane space. So, in other words, the NADH and the FADH are in here. They're in the matrix. When they get rid of their hydrogen and their electrons, the electrons are going to go down an electron transport chain, and that's going to allow energy to transport hydrogens into this space here, the intermembrane space between the outer and the inner membrane. So that's where these hydrogens are going to build up. Now it turns out that that is actually like stored energy as well. When all the hydrogen ions build up there, the only way they can flow back in is they're going to flow through an enzyme we'll talk about in a second called ATP synthase. And as they flow through that enzyme is going to, the hydrogens are going to flow back into the middle, and that's going to generate ATP. Now, the driving force of this is oxygen. So remember, we talked about how cell respiration is aerobic. We haven't talked about oxygen at all. We know that it's necessary, but we haven't talked about what actually happens to it. Well, what actually happens to the oxygen is it's waiting here at the end of the electron transport chain. It picks up the electrons. It's almost like sucking the electrons towards it. It really wants electrons. It's very electronegative. And so that's pulling the electrons towards it, and it hooks up with the electrons and hydrogen ions, and we end up with water as a final product. So the water in cell respiration, in the overall formula, if you remember our original formula, it was sugar plus oxygen. Aha, this is where the oxygen's actually coming in, made carbon dioxide and water. This is not a balanced equation. The oxygen is going to pick up the hydrogens ultimately from the glucose, really, and that's going to form water. The carbon and the oxygen that are in glucose itself, that's actually going to end up as the carbon dioxide that you exhale. So this is going to make approximately, some books will say 34. It's a little nuanced there. It has to do with transporting electrons from glycolysis. But for our class, we're going to probably just stick with 32 additional ATP, meaning we don't make a total of 32 ATP in cell respiration. We make 32 more. We already made 2 net in glycolysis. We made 2 in the Krebs cycle, And now we're making another 32 in the electron transport chain, or it's also called chemiosmotic phosphorylation, and that's where we're getting our total of 36. This is also called oxidative phosphorylation, or it's also called chemiosmotic phosphorylation, because it has to do with pumps. Chemiosmosis has to do with concentration differences on two sides of a membrane. So chemiosmotic. Phosphorylate is just a fancy word for adding a phosphate to something, by the way. So phosphorylation, they're talking about how ADP is getting a phosphate added to it to make ATP. So it's oxidative phosphorylation or chemiosmotic phosphorylation. And this little diagram is sort of showing what's happening, how NAD gets rid of its hydrogen, it becomes NAD+. The electron transport chain generates energy. They're not really showing what that energy is used for. The actual ATP is going to be made by ATP synthase. Let me show you a little more detail about this. So again, NADH and FADH2 are passing their electrons only down the electron transport chain to oxygen. Oxygen's waiting at the end, and it's actually pulling the electrons towards it. No oxygen, none of this happens. The energy that's released is used to pump hydrogen ions into the intramembrane space. So again, when the hydrogens come off, the electrons from the hydrogens go down the electron transport chain. That leaves you with hydrogen ions, hydrogens with a positive charge. Technically, those are actually protons, believe it or not, because hydrogen only has one proton and one electron. So if we take away the electron, this is actually a proton. Just FYI. Anyway, the energy pumps the hydrogens into the intramembrane space. They build up in that space, so now you have all these hydrogens, and their only way of flowing back into the middle, into the center of the mitochondria, is through our enzyme ATP synthase. And when they go back through there, ATP synthase, it's almost like a little rotor, and it spins as the hydrogens go through it, and it builds ATP. So as those hydrogens come back through, ATP is built. And then everything sort of hooks up with the oxygen at the end. So our oxygen ends up becoming water. Technically, this is not balanced either. Obviously, you need more hydrogens than oxygen. So I'm not balancing the equation. I'm just saying that the oxygen will ultimately become part of water. This diagram is showing what's happening. So there's the NADH and the FADH2. And notice how they're getting rid of their hydrogens. The electrons, this is the electrons traveling down this electron transport chain. They go to here. They go to all these proteins that actually have names, coenzyme Q and cytochrome C. You don't have to know any of that. And the power generated by these electrons being sucked towards oxygen waiting at the end is used to pump the hydrogen ions into the intramembrane space. So this is the intramembrane space. This is the outer membrane of the mitochondria. So this is this membrane in a mitochondria, not in a cell, but in a mitochondria. And then this is the inner membrane, the cristae. And this space is right here. This is the space we're talking about where all the hydrogens build up. And this is called oxidative or chemiosmotic phosphorylation. And this is ATP being made by ATP synthase. So all these hydrogens, they come flowing back through, and this sort of spins, and it builds ATP. So at the end of this process, we make water, we make a bunch of ATP. This is another diagram of the same thing. Again, inner membrane of the mitochondria. This is the electrons, NAD and FAD dumping them off. And then the hydrogen getting pumped. And then ATP synthase right here at the end, making ATP. All right. And so to summarize, glycolysis broke glucose into pyruvate. It only made a net of 2 ATP. It also makes 2 of these NADHs, and it made pyruvate. No CO2 made in glycolysis, notice. Then the pyruvate goes to the mitochondria. It gets turned into something called acetyl-CoA. You also make some CO2 as a waste product here, and you also make a couple more NADHs in this step. The citric acid cycle, or Krebs cycle, makes more NADHs. It makes FADH2, and it finishes breaking the glucose down. You actually make 4 CO2s here, you make 2 here, and there's our 6 CO2s that are made in cell respiration. Only 2 ATP. Then our very last step Electron transport chain takes all the electrons and hydrogens from NAD and FAD, and we end up making, they say 34 here, really it's 32 in most animals. Again, we're not going to get into the details of it. Bottom line, you end up with a total of 36. Some books will say 38. But almost all of them, if you're asked where are most of them made, it's here in oxidative phosphorylation or the electron transport chain. That's where almost all the ATP are made. So just another picture of the same thing, and another picture of the same thing. And we're going to skip that, and we're going to skip that. All right, fermentation, just a heads up. So what happens if there's no oxygen? Well, it turns out, remember that glycolysis, one of the products is that it took NAD plus and it converted it to NADH. NAD plus is necessary for glycolysis to continue. So if there's no oxygen, normally this NADH would go to the mitochondria and get rid of the hydrogen and get recycled. But if there's no oxygen, we've got to turn this back into NAD plus so glycolysis can continue. That's the whole reason for fermentation. So we don't make any more ATP. Fermentation by itself doesn't actually make additional ATP. Sometimes they'll say fermentation makes 2 net ATP because they're saying they're including like glycolysis plus fermentation, and they're calling it the fermentation pathway. But fermentation by itself, it does not make additional ATP. What it does make, it recycles the NADH back to NAD, and then if it's a bacterium or yeast, we usually see ethanol and CO2, that's alcohol, like drinking alcohol. The CO2 can be used to make bread rise. That's why we use yeast in like making fresh bread. And then animals make lactic acid. Some bacteria actually make lactic acid too, like lactobacillus, which is a yogurt bacteria. That's why yogurt tastes sour or sour cream. They can make these things using bacteria that make lactic acid. But in us, that lactic acid can make our muscles burn. So here's what happens here. Glycolysis happens, and notice this split. No oxygen, fermentation makes lactic acid, or ethanol plus CO2. If oxygen is present, oxidation of pyruvate, Krebs cycle, electron transport chain, etc. We make way more ATP this way. Here at the end, we've only made a net of 2 ATP per glucose, because we don't make any additional ones here, and we only made 2 up here, right? But if oxygen's present, then we go this other route. All right, and again, that's just showing, again, the difference. Alcoholic fermentation, lactic acid fermentation. Notice the goal of both is the same. It just depends what enzymes the organism had. If we had the same enzymes as a bacterium, we would basically get drunk when we exercised. We would actually make alcohol and CO2 as our products, but we don't. We have a different enzyme, so we make lactic acid, which is what makes your muscles burn when you exercise. Aerobic respiration is when you're making your ATP with oxygen. So like long distance running, you use glycogen from your liver and muscle, breaks into glucose, and then you resort to fat after that. Soccer and basketball are pretty much aerobic. Anaerobic, that's what happens when you run out of oxygen in your muscles, and that's where you feel that burn in your muscles. So sprinting, heavy weightlifting. At the end, that's why you're supposed to cool down, because your body will get rid of that lactic acid. Once you get oxygen there, it'll reverse everything. But if you don't cool down, a lot of times you'll end up with muscle soreness. All right, and the last little thing here is just explaining that you don't have to use only carbohydrates for energy. Even though the formula we learned, because that's the main thing we use, is glucose. We can break sucrose into glucose. We can break glycogen into glucose. That's how we store energy short term. We can break starch in potatoes and rice and things into glucose. That's what we go to first. But then, when we run out of that, our body goes next to fat. And, unfortunately, if you're not careful and you starve yourself, your body can also resort to protein. That's why a lot of times when people go on a diet, they go on one of those high protein diets. It's so that your body will burn fat. You eat no carbs, you eat a lot of protein. Your body will burn fat, but you won't lose muscle or protein, because that can be very dangerous. Bottom line though, your cell can use pretty much any food you eat to make energy. Carbohydrates will go through glycolysis as their first step. Fatty acids will go straight to the acetyl-CoA, and proteins will get broken into amino acids, which will feed into